Good morning, everyone. My name is Tutuzile Kupega. Welcome to Life Sciences Grade 12. Today's topic is homeostasis. So last term, we covered the topics before that, right? In the endocrine system. And now today we are dealing with homeostasis. Remember I said we are done with revision now. So now we are moving on with our topics. So this stems from the topic of endocrine system, but it flows to homeostasis. Up in the front here are my details. If you have any questions, feel free to send me an email if you are struggling with any topic, anything at all related to life sciences. Ne? And then at the end of this lesson, we should have discussed homeostasis and the overview of homeostasis. So the overview of homeostasis, we know that all cells in a multicellular organism are surrounded by a liquid called tissue fluid. So all of the cells are surrounded by this liquid. Tissue fluid makes up the internal environment of the body. It consists of blood plasma that is leaked out of porous capillaries. Tissue fluid consists mainly of water in which various substances are dissolved. And then the makeup of tissue fluid changes continually while body functions and metabolic processes are taking place. And then you'd expect that to happen, of course, because let's say one day when different things happen to your body, sometimes you eat certain foods that would change the tissue fluid. Sometimes there are different types of nutrients released in your body, different types of substances released into your body. That's why the makeup of this tissue fluid will constantly change. Let's say you're taking some medication, that would also have an effect on your tissue fluid. So that's why we're saying it's continually changing because of these body functions and metabolic processes which are continuously taking place. Then cells take oxygen and nutrients such as glucose from the tissue fluid. So this oxygen and the nutrients that we need, they are in the tissue fluid. The cells then take these nutrients such as glucose, for example, from this tissue fluid to use it for the internal important parts. Rather, maybe the cells of the muscles will take this glucose as it will provide it with energy, different things like that. Tissue fluid carries away waste products, for example, carbon dioxide, urea, and excess salts produced during cellular processes. So there's a lot, if you can see, there's a lot in this tissue fluid. So waste is also carried away here. And also nutrients are also carried away here. So a lot of things are happening here. Waste products are excreted from the body by the excretory organs such as the lungs, kidneys, and skin. You know that those are the excretory organs, right? The lungs would be responsible for giving out the deoxygenated blood, and then the kidneys are involved in the urea process. And then the skin, through sweat, you can be able to release all this waste product. And this is the typical diagram of if you're asked to draw, just to visualize the tissue fluid and these body cells. So this is the tissue fluid in yellow here, and these are the body cells. So that's how it surrounds it. So that's why, so you see, absorption is easy like this because the cells are already surrounded by this fluid. So they can easily absorb all of this fluid. And then an optimal internal environment has to be maintained for body enzymes to function properly. However, this optimum level is not exact. Conditions can fluctuate within a narrow range, but the body must take steps to ensure that the range is not exceeded. When enzymes are denatured or destroyed, the metabolic reactions under their control will slow down or stop altogether. And if the enzymes are not working properly, cell function will be negatively affected and cells may die off. You know this, you need certain enzymes to facilitate with certain parts of your bodily functions. So if this internal environment of the body is not kept or not maintained to its optimum performance or position or condition rather, then the enzymes would be affected because enzymes need to exist in specific conditions. So if a specific enzyme is responsible maybe for food breakdown, it needs to maybe exist in certain pH levels. It needs to exist in certain temperatures, things like that. So if you alter this internal environment, you affect those enzymes which need to exist in specific conditions. And when you affect the enzyme, then the enzyme could either be denatured or destroyed. And when you denature or destroy this enzyme, it means all of these functions, the metabolic reactions under the control of this particular enzyme will be affected. And then that's when, if you affect those particular bodily functions, the metabolic reactions and so on, then you ultimately affect the functioning of the cell functions there and the whole body. This continual monitoring and regulating of the internal environment of the body is called homeostasis. The word homeostasis comes from a Greek word that means to stay the same. Homeostasis is defined as the maintenance of a constant internal environment within an organism. So you remember when we did the endocrine system, we discussed that the point of the endocrine system is to maintain order, to maintain balance. So that's exactly the same thing that's happening with homeostasis here. It's just that with homeostasis, we are not only focused on the endocrine glands, but endocrine glands are part of homeostasis. You understand how it works, right? In the endocrine system, we're focusing on the endocrine glands. But now we are maintaining balance in the whole body. We are discussing that maintenance of balance in the whole body, which is inclusive of endocrine system, but other systems and other glands as well. Okay, hi Kyle. Guys, I've missed you so much. Welcome back to class. It's so good to see you. And then next up, in order for the body to function at its optimum level, various factors associated with tissue fluid have to be kept at a constant level. These factors include what? Carbon dioxide concentration, the oxygen concentration, mineral salt and water concentration, urea concentration, glucose concentration, pH, and temperature. These, as you've noticed, they affect your bodily function, so they need to be kept at constant levels. If you have too much carbon dioxide concentration in your body, it won't be good for you. Then it could likely lead to death because it would affect your respiratory system, as well as the oxygen concentration. If you have too little oxygen concentration in your body, then your body can't function optimally. The mineral salt and water concentration would also affect a lot of other factors in your system. Urea concentration as well. That's why you need to regulate the water intake, and the kidneys need to function optimally. So if this part here is affected, most likely the kidneys would suffer, and then ultimately your whole body will suffer. That's how the body works. You touch one part and you affect it. You hurt one part, you hurt the whole body. It's connected like that. The glucose concentration, like we've learned with what's this, diabetes, it would affect that, because if your glucose concentration is too high, then you can get diabetes. If it's too low, then that's also still a problem because your body needs the glucose concentration. Your pH level, again, certain enzymes need to function at a specific pH level, as well as other bodily functions need to function at a specific pH level. So if it's too alkaline or if it's too acidic, then that's still another problem. As well as temperature. Human body's temperature needs to be maintained at constant levels, guys. You'll notice now with the biggest example, everywhere when you go to the malls, you go anywhere, your temperature is taken. Why? Because when something is off in your body, if you have a certain infection or whatever, it will affect your temperature, and it is telling. So if your temperature is too high, it could tell that the internal something in your body is not going well. You might be having an infection. So that's why we are getting our temperatures tested everywhere. When your child is sick, you can look just by the temperature, and then you can tell that this baby is sick, they have a fever, because the temperature would be too high. Also, when people are the dead corpses, the corpse, of course, is a dead person, but the temperature there, it really now it lowers way below zero. So temperature is also a specific and a good indicator of one's health. So these ones need to be maintained. So when we discuss homeostasis, these are all the factors that need to be kept at constant level, and we will need to learn how they are regulated. Many organs in the human body are involved in homeostasis. Many of these organs, let me try highlight as I go. We have what? The lungs. Lungs do what? They help to maintain constant concentration of oxygen and carbon dioxide in the blood. We've just explained that. And this part here you find in the respiratory system. You also have the kidneys. Kidneys help maintain a constant concentration of waste, salt, and water in the blood. And it is involved in the urea system, or the excretory system. We have also the liver and glands in the pancreas, which help to maintain a constant glucose concentration. Then we have what? The skin, which helps to maintain a constant body temperature. We also have what we call the brain, which is also involved in homeostasis. The brain controls the homeostatic process by sending instructions to the organs. And you'll remember again that a lot of the hormones are excreted firstly from the brain, or the glands in the brain. Then the glands in the brain would then be the ones that would regulate the release or the excretion of other hormones to facilitate in this process. Then hormones secreted by endocrine glands are also involved in sending messages to organs to control their functioning. So for those of you who attended class and even watched YouTube videos when we discussed the endocrine system, this might feel like revision to you, because there are things we covered like that. And then this is a graphic representation of organs involved in homeostasis. You've got the liver and the pancreas over there. You remember the pancreas is the gland of the endocrine system, and it is involved in the release of these two hormones, insulin and glucagon, and it works closely with the liver. You have your lungs, which regulate what? Carbon dioxide and oxygen concentration. You have the kidney, which regulates the process of urine. So it is involved in the water and salt concentration in the body. Then you've got your skin, which not just, remember I mentioned the sweat. You see what happens here, it regulates your temperature. Skin plays a very big part in temperature regulation over here. And then, yes, those are all the processes. With the insulin and glucagon, they maintain the glucose concentration in the body. Then this here, remember the diagram of the body cell and the tissue fluid? So that's how they work together. These are all the organs involved. Not all of them are listed here. We have also the brain, which is not shown here. And then negative feedback. You remember negative feedback, right? In order to maintain a constant internal environment, the human body has a number of regulatory mechanisms. These mechanisms regulate themselves. For example, if too much of a particular substance is present, there needs to be a way to regulate production of that substance and reduce it. Similarly, if there is too little of a particular substance, steps must be taken to increase its concentration, such as self-regulating mechanisms called negative feedback. You'll remember negative feedback. It is the product of that particular reaction is the one that gives feedback of whether or not to increase more or to reduce the concentration of it. A negative feedback system has three parts. We have the receptor. There is a receptor that detects the move away from the normal concentration or state and alerts the appropriate control center. And then you also have the control center, which processes the information and initiates an appropriate response. Then you have the effector that corrects the imbalance and brings conditions back to the normal concentration or state. So you need to remember these three in the negative feedback. You have the receptor, you have the control center, and you have the effector. So as we're going to discuss different types of the negative feedback systems in the body, we will also need to be able, when we discuss that, you need to be able to identify which one of it is the receptor, which one of it is the control center, which one of those ones is the effector. Like we said, the receptor is the one that detects the change, that now it's no longer normal. Now it's either too high concentration or too low concentration. The receptor detects that. Control sector. The control center will then be the one that's deciding, okay, because the concentration of glucose is low, let's release enough of the glucagon. Or because there's the high concentration, then let's release insulin. Examples like that. And then effector, the effector is the one then that is released. So for example, if we had to release more glucagon, the effector would be glucagon, which is now going to correct the imbalance. So this is the typical diagram, like I've said. This is here, you have, when on this first part here, you have normal concentration of factor in the body. Then this factor will increase. Effector, which causes the corrective changes, will then be released there. And then because the effector was released to correct these changes, then the factor would decrease back to normal concentrations. But in the case where normal concentration and there's a decrease of the factor, the effector will then be released to ensure that there will be an increase of the factor, and then the body again is back to the normal concentrations. And then we will discuss now the negative feedback in regulating the blood glucose concentration. The concentration of glucose in blood must be carefully regulated at all times. Too much glucose in the blood can cause water to move out of cells by osmosis. And then too little glucose can leave one feeling weak and dizzy. The blood glucose concentration is regulated by two hormones produced in the endocrine cells in the pancreas. You remember those the hormones that I'm talking about? It is the glucagon and the insulin. Also this year, when they mentioned that too little glucose can leave one feeling weak and dizzy, you will remember that whenever, let's say, you feel a little dizzy, let's say you get to school and you feel a little dizzy, they usually ask, did you eat today? You know? And then that's why they would say you have low blood sugar, because when you eat, your body gets this enough glucose boost in your body, and that gives you enough energy. So you'll start feeling weak and dizzy if you probably have missed a lot of meals. That would happen, you know. Then the concentration of glucose in the blood, we know it, that's why it needs to be constantly regulated, because it can affect you, and the reaction of it is very quick. One hormone, which is insulin, like we've mentioned, it lowers the blood glucose concentration by stimulating the conversion of soluble glucose into insoluble glycogen in the liver and muscles. The other hormone we know and we've discussed is glucagon, which has the opposite effect and causes glucose to be released into the blood. Insulin and glucagon have opposing effects, and therefore they are antagonistic to each other. You'll remember this word here. It means they act Their actions are against each other. If the one increases the concentration of a factor, the other will reduce the concentration of the factor. So that's why they are antagonistic. That's how we term the word. And then next up, this is the diagram to show you typical negative feedback mechanisms of the blood glucose concentration. You have normal concentration of glucose. You'll notice, as long as you understand the very basic structure of negative feedback mechanism, you'll just substitute when you're dealing with blood glucose concentration, you need to just substitute the correct factors, the correct effector, then you're good to go. So as long as you understand those, all of them are typically like this, all of them. You just need to understand the different factors and effectors involved in those systems. So that's the clue here, the hint that I'm giving you. When you study, make sure you understand clearly in each of these different negative feedback mechanisms. You need to understand which one is the factor and be able to identify which one is the effector. If you know those two and you just know the basic structure of a negative feedback, basic diagram of the negative feedback mechanism, you can just substitute there. Like here, it's normal concentration of glucose in blood. Let me compare it with that one I told you. Here, it's normal concentration of factor in blood. So it means the glucose is the factor. So it means here, when they say increase of factor, when we're dealing with blood glucose, it means it would be increase of glucose. And here it would be decrease of glucose. You understand? And then when there's an increase of glucose, which effector would be released? We have noticed that the effector that will be released is the insulin, which is released by the pancreas. But if there's a decrease in glucose in the blood, then glucagon will be released because glucagon plays a part in increasing the glucose concentration. But insulin plays a role in decreasing the glucose concentration, which will bring the body back to normal concentrations of glucose in the blood. So understand the factor and the effector, and you're good to go. Then the negative feedback in regulating carbon dioxide concentration. Carbon dioxide is a toxic waste product of cellular respiration. Carbon dioxide diffuses from the cells where respiration takes place into the blood. The blood then transports carbon dioxide to the lungs. It is removed from the body during exhalation. When you exhale, you remove carbon dioxide. When you inhale, you take in oxygen. The carbon dioxide dissolved in the blood turns the blood plasma and cerebrospinal fluid acidic. So it changes the pH concentration if it changes, right? The carbon dioxide changes the pH level, which means what does it do? The pH will drop. And then when the medulla oblongata senses the lower pH level, it responds by initiating deeper, faster breathing in order to reduce the carbon dioxide concentration in the blood. Once the normal concentration of carbon dioxide is achieved, the breathing rate returns to normal. And if the concentration of carbon dioxide is low and the concentration of oxygen is high, the urge to breathe is less and the rate of breathing will be slow. The medulla oblongata, I've noticed right now, only responds to the carbon dioxide concentration and not the oxygen concentration. And this is a diagram here. You have normal concentration of carbon dioxide in the blood here at the center for both. This is normal, né? But now this is normal and suddenly there's an increase of carbon dioxide. Then what will happen? Remember the medulla oblongata responds to carbon dioxide concentration. So it stimulates the increased breathing. When there's increased breathing, then there'll be a decrease of carbon dioxide. But should it happen that there's a decrease of carbon dioxide in the blood, it will be decreasing to levels lower than normal. The medulla oblongata does not cause increased breathing. It will just stop and not do anything. Then there would be an increase of carbon dioxide in the blood. So you understand how it works, right? You've seen this diagram. They're really, really all similar, as long as you learn to note the differences. So, yep, that is it from me today. We will finish up on Monday. And then as soon as there's more of you guys in class, we will do Kahoots. But for now, we'll just carry on with the curriculum. Are you guys okay? Any questions that you may have, feel free to ask me now. You're welcome, Kyle. Hi, Busi. Are you guys okay? It's nice seeing you guys too, Kyle. All right, Busi, I guess you're good, ne? Okay, guys, I will see you on. Uh, really, you're writing a test. What are the topics, Kyle? Are you ready for the test? When are you writing the class test? And I'm pretty sure you were ready for it. Can I let me allow you to speak actually? Yes,